Welcome, everyone. And I first, before we get into details, want to pay my respects to our elders, past, present, and emerging, as we are here on Whadjuk country. And when you think about the deep history and long time of this place, I think David in his talk is going to also give you a little bit of a window on how we better understand these old places and seascapes. Housekeeping, phones off. If sirens go off, flee the building straight out the way you came. I'm not sure where the marshalling point is, and washrooms are around on the other side in case you need them. So my name is Jessica Meeuwig. I'm the Wen Family Chair in Conservation at the University of Western Australia. I'd like to very much acknowledge the Wen family who've made the chair possible in perpetuity. So once I pop off the perch, awesome, there'll be another one. But it's a really wonderful thing when people like the Wens contribute to universities in this way, and I won't steal Amit's thunder on that. So my other job is to simply introduce the Vice Chancellor, who's going to talk a little bit about the Wens and the importance of this lecture. Then I'll come back and I'll introduce our speaker.
Kaya Wangju, hello and welcome. It's a great pleasure for me to welcome you all, and I'm delighted to see so many students. When I see young people, I just assume that you're students. Even if you are not, we are all lifelong students, so pretend that we are all students are going to learn a lot from this lecture. But before I go any further, let me welcome our distinguished guest. And I just managed to speak a few words with him en français. So, Monsieur, je vous souhaite bienvenue à notre l'université de Western Australia. One of the benefits of being able to greet somebody in his or her language is that you build a very good relationship right get-go. And I have been impressed with French speakers in general. I learned English from primary school all the way through now, but no English-speaking gentleman, lady has ever complimented me for my language. Every time I speak French, I get that compliment. And our distinguished guest is no different from that. So I'm digressing because I see so many young people, young students here. I speak 5 languages, and I'm very blessed to have had the opportunity to learn those languages. I encourage you to take every opportunity you get to learn language, because language is a tool. Language is a tool not just to communicate. Language is a tool that opens my mind, because if you don't have the tool, you can't open your mind. So I like Europe, but the country that I relate the most is really France. And why is that? Because I speak the language, I understand the culture, I can enjoy, if you will, the nuances of French life. So that's just one example. So let me then encourage you to keep learning and bring it to a modern level. I was trying to encourage my son, second son, we are Canadian, to learn French. I said, well, Dad was not born in Canada, but I picked up French. And you are Canadian, Canada is a bilingual country. It's a shame that you don't speak the language. So he graduates from the university. I wanted him to go to Quebec to take a French immersion course, which is very good, that my university used to run. So he would not do it. At some point in time, he gets frustrated. Now he landed a job with a bank. So he said, Dad, you don't understand. So we learn languages, but they're different languages. I said, tell me which languages they are. So programming languages, some advanced level. So I do acknowledge that you learn other things that my generation perhaps you haven't learned. So coming to the lecture, let me echo what Jessica was conveying to you, and you would have been very welcome to steal my thunder. But since you haven't, so I'm going to acknowledge the wonderful gift that May and Chi Wen endowed for the university. So the 2 aspects to the gift. One is just the concept of an endowed chair. If we go back to the founding of this university, we had great visionary leaders who founded the university. Not only they founded the university, they had great visions that inspires us today, certainly inspired me today. So one of that vision was that this university would be one of the very best that it can be. And one way of doing it is actually to support scholarship, search for knowledge, seeking wisdom in perpetuity by endowing our first endowed chair 111 years ago in agriculture. So that chair over the years, the chairholders of that chair, the Winthrop Hackett Chair in agriculture, have made enormous contribution to the agricultural research in our state. Without the contribution of that research initiative, actually our agriculture would not be where it is today. So that's at the time of our founding. So I consider the generation of Sir Winthrop Hackett as our founder's generation. And I consider the Wens as part of our builder's generation. So they continue to build on the legacy that Winthrop Hackett left behind. We kind of ignored doing the sort of things that are important for us to do. The example Hackett set for us for a while. So we have picked up the pace, and one of the early chairs have been endowed by the family. So they not only support us, they support many, many, many other causes through their family foundation. And that covers from environment to health to other aspects. And in this case, of course, we are interested in health of different kinds, health of oceans. And they add to our existing strength in oceans research, trying to understand the oceans better, and more importantly, perhaps, trying to protect the oceans and whatever unknown resources we have, living or otherwise in our oceans. What differentiates the Wens from other gifts that we have received? Of course, we're grateful for any gift that we receive. Without those gifts, we would not be the university that we are today, because our funding model is such that we can make do with our routine things, but for us to build excellence, we need additional support. Our business model simply doesn't allow for that. So there comes philanthropy. So what differences they went from the other philanthropists? They're engaged philanthropists, particularly May. Okay, from Jessica's point of view, that may be a little bit more challenging because she has to deliver.
It's awesome.
But from impact point of view, that engagement ensures that he's not only, seeking knowledge, is making sure that the knowledge that we gain are applied, that they have impact. So that's impactful, if you will, philanthropy. So I cannot thank you enough for placing your trust in your university. Certainly Chichi is a graduate of the university. A number of their children are graduates of the university. So I claim that this is their family's university. But nevertheless, people who have the good fortune of having resources, what happens is they work very hard to generate those resources. But they have to work even harder to give away those resources so that they have impact. So it's hard work. Believe me, giving money actually is quite hard work, and May knows that very well. So thank you for placing your trust in us, because you could have given your support to so many other organizations. We are very grateful. So I hope that those of you who are attending the lecture will not only enjoy the lecture, but you'll learn something from the lecture, and you'll go and reflect what you have learned, and as will I, and then try to put some of the lessons that you pick up from today into your thinking as to how you want to make your own contributions. Those of you who are interested in oceans, saving oceans, those of you who have general interest like I do, just how we advance knowledge. So with that, let me turn the podium back to Jessica to introduce our speaker.
Thank you. So thank you, Vice Chancellor. Before I introduce our speaker, I also need to acknowledge the other people who support this public lecture. This is a really important part of the chair. It's actually in my contract that I have to find at least one fascinating person a year and bring them from wherever to Western Australia to give this public lecture. And that, I think, is part of how this chair has been designed. But I also want to acknowledge the support of the Oceans Institute, the Development Office, the Forrest Foundation, and Jock Clough Marine Foundation, who offered you the glass of wine and the nibbles before you arrived. So by collaboratively all working together, we can leverage to bring really interesting people here to come and share their knowledge and their experience with us. So on that note, I'm going to tell a short story about David Mouillot. So I've known David for about 10 years. We first met in London, and then I was very fortunate to go out to the University of Montpellier before COVID for a fascinating workshop that has generated a number of papers, one of which was published in Science last year, and another one that came out in another top journal with us on protected areas. But I was stunned to find out when David and I were chatting this weekend that he got his master's at the University of Corsica and his PhD. And the reason he went to the University of Corsica, which is one of the smaller universities in France, is because he didn't have very good grades as an undergraduate. And for me, that was a wonderful, he approved my including this in his bio, right? So I asked him first before I publicly embarrassed him about his poor grades as an undergraduate. But the reason I thought that story was so important is that we all have different pathways to what we're going to choose in our journeys. And some of those don't fit in conventional terms of a 4.0 GP grade point average or this or that. And I know I've got some wonderful people in my lab who, on paper, didn't look like they should get a scholarship, right? So for me to hear David's story that he went to this small place and then he started thinking about nature, he was introduced to thinking about marine parks, he was a math and stats geek, his words, and then he's evolved into somebody who grabbed opportunities in life very early. So he's been a professor at Montpellier, and moi je parle français aussi, but with a Quebec accent, which means the French don't actually think I speak French. But you know, from there, he's grasped opportunities very early. So David's not talking about environmental DNA this afternoon, but he was one of the early explorers of how do we take buckets of water and identify what species are there. And he and I are going to go hunt for coelacanths, right? These old shark fish. But also he was one of the first adopters around understanding the opportunities presented by artificial intelligence and machine learning and these kind of things, which is the subject of his talk tonight. But what I will say, this will be a relatively technical talk. I've seen it. But it's going to give you a window onto how machine learning is going to transform our ability to understand our oceans, which are so vast. It's pretty hard if you're just doing it manually. So with that, I'll turn it over to David.
Thank you, Jessica. Thank you, Vice-Chancellor. So, all is said about me. So, I'm also holding a chair in France about environmental DNA to seek the most endangered and hidden species in the sea. But today I will talk more about where and why species occur and at which abundance. These questions are very central in ecology, and we all know that they are based on field observation, as you can see in Australia for acacia. And we still try to relate these observations to environmental habitat variables. In the sea, we know that most big animals, that vertebrates, are declining across time. And we know that under fishing pressure and climate change, we have some explanation to this major decline. So I will have two parts in my talk. First, I will show how we can improve such species distribution models, how we can be better at filling gaps, because we cannot sample all time, everywhere. So we need to get data where we cannot provide field observations. And I will also show how, using these new models, we can better understand the links, the causal links between species, air, and the socioeconomic context and the conservation efforts, particularly in the oceans. So the basic of predictions are very simple. We have some observations. We have some layers representing environmental conditions. And we just know that where the species are present we have positive associations, and where the species are absent, we have negative associations with such conditions. And the models are very simple somewhere. They just predict the suitability of certain conditions to the presence of a species. So it's very, very basic. And using such models, we do great things. We can predict, for instance, the future of many species. We can develop an index of risk of marine life under future conditions. So let's take most of the species in the sea. We can predict future conditions of environmental variables like future temperature, oxygen, and we can know whether the species will survive in good conditions or not. It has been a great paper assessing a risk index for most species in the sea. And we can provide as well with very pessimistic predictions where we can see the increase or mainly decrease in red of biodiversity across the oceans according to the new scenarios of climate change, here the RCP 8.5, which was the pessimistic scenario some years ago and which is the most expected scenario by now. And we can see that the tropical countries, the tropical seas will see a major decrease in species richness in the coming years. But this model have some limitations by now. The first is that species distribution are poorly known. Why? It's because that less than 5% of the ocean have been sampled. It's very, very little. When you see this map, only the red and the blue areas have been sampled till today. This map is from yesterday, so it's very new. So we know that we have many uncharted territories in the sea, so we still poorly know the distribution of species according to environmental and human conditions. So we need expedition and global survey with standardized protocols across the ocean. I like this map as well. It's the view of explorer, natural explorer on terrestrial ecosystems. So you see the proportion of samples according to the effort, and you can see that it's totally disproportional, with most of the effort carried out in Europe, in the US, in Eastern Australia, and many continents and coastries are totally undersampled. There is no data in many countries like Madagascar, Bangladesh, why not, or other countries where we lack data. So it's why I collaborate with Jessica. I took this picture some hours, some days ago, a crazy one, crazy map that many guys of our lab know. So it's why I collaborate with her, because she's the only one to go to these places, very isolated places, to collect more field data, to complete the species distribution that we only poorly know. Another limitation is that by now, most of models consider species alone. We tend to model species one by one. It's what we do in many fisheries stock assessment, for instance, in most of modeling. And we know that species interact. We know that species are linked by behavior, are linked by predation links, etc., etc. And we know that predicting a species alone is a very limited view. For instance, I can show this article, choose one of them. Some people from Perth are doing that. And they show that when they do experiments, they look at these mesopredator fish like fish between 10 and 20 or 30 centimeters, and they look at their behavior when a diver or a shark is coming. And it's very clear that when a turtle is coming, we have some behavior, some fly away, some have no response, they don't care of the turtle, some turn and some hide. So we can see that when a turtle is coming to the game, when the turtle is via assemblage, some fish cannot be spotted. So we clearly see that when we do surveys, if we not include the turtle, it's hard to predict the fish. It's even worse with sharks. When a shark is coming, we can see that most of the species fly. So it means that when we get more sharks, we get less species in the samples. It's what we get in marine reserve. We tend to see less species when the sharks are there. It's because most of the species fly. So all the species interact, and if we do not predict all species together, we miss one big part of the puzzle. It's another limitation. We tend to predict species according to very, very local conditions. When we try to predict the presence of plants, we look at the soil and very local conditions like weather, precipitation. And we tend to do the same for marine species. But we omit that most of marine species, compared to plants, move, they have large home range. And if we try to predict a species according to one habitat, we missed as well many aspects. That the species can connect between mangrove and coral reef, that the species can be safe on the shore but caught by fishermen away. So the goal is to try to move from local conditions to a view embracing the seascape, a broader view of conditions for fish. This is the case for marine protected areas. I've done that for many years, and we try to predict fish presence or abundance according to a very binary mode, which is we are inside or outside. My students always ask me that. Is the sample inside the protected area or outside? And we know, let's take this example in Mexico. They perform transects inside a partial protected area in orange, some no-take in red, and they have control sites outside. And they try to predict fish abundance according to this binary variable, which is very simple. Are you inside or outside? And we tend to see this kind of patterns where you get more abundance outside than inside. Okay, great. But at which distance are we? Can we integrate all these conservation efforts into a seascape instead of being just inside or outside? Can we include the fact that the marine protected area has some partial one outside, that this one is north, this one is south, and I think if we try to include such patchy and complex habitats, we'll be better in predicting fish abundance and species. If we look at sharks in marine, protected marine areas, the out and in has no sense as well, because we know that most of species of sharks move a lot. They have large on range. And we look at if we want to protect a large portion of activity of the sharks, we need to protect at least 75 or 100 kilometers of reef. We need very big MPAs to protect the sharks. But a distance doesn't mean anything in terms of habitats, because if you place your MPAs there or there, it's not the same habitat. So we feel that we need to integrate all this complex seascape into modeling to avoid the fact that in one place there is no fishing, in another one there is fishing. We know that there is regional fishing, even if you are in no-take area. So sometimes in your academic career, you have to attend committees, PhD defense, you are invited in many occasions. It's good to escape, you try to avoid many traps, but sometimes you can't. And it was, I think, 3 years ago, I was invited in a PhD committee about orchids. I didn't want to go, but we have to try to help colleagues. So I said, yes, let's go for an orchid committee. I thought it would be so boring to hear about orchids and artificial intelligence, to feel I'm not very good at. And it was, I must admit, one of the big shock of my life, for my career research. Because what this guy did, it was a PhD student, 23 years old, and his title was, I will map orchid assemblage at a global scale at 1 kilometer resolution. I say, he smoked something or something very strange. So let's see what this guy is telling me, that after 1 year, he got a global map of orchids assemblages. What I like is that he provides rapidly these kind of maps showing where the models are. And I am more specialist of GLM, classical statistical models, or decision trees or machine learning model or Maxent. And this guy goes to these deep learning models which are a bit like black box, but much more accurate in terms of predictions. So they are just based, for instance, on all what we know, on ImageNet, for instance. How can we recognize species according to pictures? It's very easy. It's based on stripes and colors and many things. And as an output, you know that it's likely to be a zebra. It's very easy. So we saw that everywhere. Your cell phone is very good at recognizing you. So we saw that in medical science as well, where you can see the lungs and you can know whether you have pneumonia or not. So it's a very binary response according to your photo you can get from medical science. And these guys told me it's the same thing for orchids. Okay, same thing for orchids. Yeah, he's told me that you take satellite pictures and I can tell you whether there is an orchid or not, whether a species, a given species is present according to just the satellite image. He was just throwing away, breaking 100 years of plant science in 1 second, telling me that only satellite pictures can tell everything about climate, soil, combination of species, and so on. And he was quite convincing because I tell him, how do you know the soil is good for orchids? He tells me, if the soil is good, you will have certain type of trees. And my model knows the trees, and you will know that orchids can be associated with tree. Okay, what about climate? How do you know that climate is good for orchids? If you have these green leaves in spring, it means that it's a temperate climate, it's good for orchids, and so on and so on. So he told me, it was very convincing that using the satellite images, he could predict orchids. And in addition to that, he told me that his model is good at linking orchids to images, but orchids to other plants and trees. So these deep learning models are good to make links between images to species and species between them, to include all that into predictive models. So this guy did that. So he used close to 1 million occurrences of close to 14,000 species of orchids. What a massive job. You can see that on average, most of orchid species have very few observations. Most of them are very rare. So the challenge is huge to predict species with so few observations. And what he did is that he used these images, and his predictive input is covariates with these crazy photos from Afghanistan near a bed river. Yes, how can he tell you where we can have orchids? Another one is South Russia. You can see there's infrared and other images in 1 year. So he took 1 satellite images per month during 1 year before the observation. And using this photo, it was good to build the best model ever predicting orchids, presence, and assemblages. And since we have photos everywhere, since satellites go everywhere, he was able to provide these maps at 1 kilometer resolution. So this guy was able to predict where the orchids occur with much, much better accuracy than all the models before at 1 kilometer resolution. And he was able to predict as well IUCN species of orchids on the map below. So total species richness and IUCN species richness. So I skimmed to downscale and to know whether he had some good result of protected areas to tell to him maybe your model is totally silly because you should have more orchid species within protected areas. So this guy extract me some nice maps from Sumatra. And he told me, with my models, I got 3 times more orchid species inside but outside protected areas. And he asked the guy from Sumatra to verify that where he predicted orchids, they were, and they were. I don't say the model is perfect, but the model is more accurate than all what has been done before. I see the big eyes of Jessica telling me it's a talk on marine science. I go back to my fish. So I was very jealous of this guy and this team working on orchids and providing global maps. I tried that all my life, and this 30-year-old master student PhD student did better than me. So I went back to my fish. I didn't want to start a global scale. I was a bit scared. I tried to look at my Mediterranean fish species and to use my new reef life survey data. Reef life survey came to the Mediterranean some years ago. with very standardized protocol that you know all, that divers counting fish on transects of 50 meters long. They are applied everywhere, the same protocol, so that's great. They have been sampling almost in every location, but there are very few samples in the Mediterranean Sea. So in my mind, say, okay, we have very few samples in the Med. If I can build up a model like this, like the one of orchids, I will be able to predict my fish across the Mediterranean at very low cost. To be great. And it's a very challenging task to predict fish species abundances. So I wanted to try not only species presence, but to try to predict abundance of fish species in the Mediterranean Sea. Because all these fish species interact, some are in big groups, some are very rare, so it's very hard to predict their abundance. According to some habitat conditions, like reef conditions, reef substrates, and some as well conservation efforts between open to fished, restricted or no-take area. So I have a good team on AR. I took the code of this guy working on orchids, and I try to predict my abundance of fish on my 200 samples in the Mediterranean Sea. We had only 47 species, so we started very modestly. So what I did, instead of having very local conditions, Mediterranean landscape, I just extracted satellite images where the fish were counted. So it's very easy. We can go to the past. Even if the fish were counted 3 years ago, some last year, I got images of primary productivity, temperature, depth, many environmental variables and habitat variables, and as well MPA, the MPA context of my fish. So we know that random forests are providing some good results. So this is the baseline. So on the y-axis is my predictive power, and you see the benchmark, the minimum baseline is what my random forest, my classical models are doing. And my first try was a bit depressing. This guy was able to predict orchids everywhere in the world. I am not able to predict abundance better than my random forest models. So I went back to this guy and I told him, why, it's so good for orchids, not good for my fish. And he told me, where are you going with 200 samples? The backside of deep learning models and artificial intelligence is the bunch of data you need. These models are very demanding. The training of the model is very demanding. You cannot do anything with 200 samples and 47 species. But he told me what you can do is that your phone knows you, not because he has many pictures of you, but it was trained on ImageNet. It knows how to recognize cat, dogs, zebra, and everything. So this model has been trained on billions of images, and then they use this trained model to recognize your face just based on few photos. We call that the few shot. So this foundation model are trained on many very big data sets, and they are fine-tuned to very specific tasks with very few images. So he told me, do the same. In the sea, you have many observations, many occurrences, and you can pre-train your model on all various occurrences, and then fine-tune your model to your specific task, which is predicting your fish in your samples. But what we did, because we have some observation in the Mediterranean Sea, so I collected only recent observation of fish occurring on the coast, and I used these fish occurrences from GBIF to train a first model, just trying to predict biodiversity. Where are the fish according to satellite images? So I use this model trained with 60,000 fish occurrences, and I use this trained model fine-tuned to my new task of predicting abundance. And then I got a major result, which is predicting power increased a lot. I got much better result when I use these pre-trained models on my small task, which is just predicting abundance across 200 samples. And the good thing is that not only this model is good on average, But this model is very, very good on rare species. So you see on this graph on the right, when the model, the random forest model, the old one, some years ago, the old one is better, it's going right. When my new model, new fancy model, CNN is better, it's on the left. At the top, I have the most common species, Coris julis, occurring in 98% of the samples. So the species is everywhere. And at the down, you have species occurring in only 2 or 3% of the samples. So very rare species. And you can see that for common species, if I use the old or the new fancy models, I get similarly the same predictive power. But as soon as I go to the rare species, my new artificial intelligence model is much, much, much better. When we have very few occurrences, my model is good because there's fish species, most of them are piscivore, large orange. They are much better predicted because I include the seascape and because I include all other species. Because we have many, many dependencies between species. So the species at the bottom are better predicted because I'm good at predicting the others as well. So I predict the whole assemblage instead of doing species by species. So it was 10 years ago, the kind of map we got in the Mediterranean Sea. You see the gradient. And with these new models, you can see what we can get on the French coast. Very much better models. So what are the take-home messages from this first part? These new models can overcome 2 main limitations, which are, can we integrate species associations? We can indeed predict all species together to predict more realistic assemblages of species. And we can as well take advantage of a seascape. It's not only because you are inside a non-protected area that you are good, that you are abundant. It depends on the external conditions, whether you have some fishing pressure outside, whether habitat is good, whether there is a mangrove very close by. These deep learning models should be trained or must be trained on over big data sets. So these foundation models need more than 1 million observations. And who is doing that in the sea? Nobody. I know perhaps 5 researchers in the world doing that.
I have 1.1 million.
Exactly. So this is the legacy of Jessica, because without these observations, we cannot build this foundations model. So without all our work and our team, thank you guys, we cannot do, us modelers, anything, because we need such huge data to pre-train the models in order that they can become accurate on small tasks, like predicting by small Mediterranean fish. So then, can we understand something? Because the backside of deep learning models, we all know that, is that it's a black box. You have some images as inputs and some prediction as output, but nobody tells you why you have a pneumonia, whether it's a zebra, whether there is orchids. They just tell you it's there. So the goal is to be better. So if we look at this, it's very easy to know whether the model is correct or not, accurate or not. It's much more challenging whether this model can tell us if MPAs are good for fish. We know partly that, but it's much, much difficult. And I must admit that maybe we have some social scientists here or medical scientists, that ecology conservation is lagging a lot behind social science and medical science in terms of causation. We are not so good enough for causal relationship. Perhaps because medical science need to be sure that a medic is doing something, that someone investing money into some social action needs to know exactly the impact of that. In ecology, we are very weak on that, and we must be better on causation. So what is the real gain of biodiversity when we establish a new protection? So I love this book of Pearl, and it has 3 levels of causation. You can see that on the scale. So the bottom level is only association, just seeing. Do we see more fish inside protected areas than outside? The second level is intervention. It's doing. After the creation of the MPA, do we see an effect? Is there any causal relationship between the creation of MPA and the level of biodiversity of abundance? And the last one, counterfactors. Imagine, so to know whether all the conservation effort we have done in the last years had an effect, it's good to imagine a world without this conservation effort. What would be the status of species without MPAs? So this is based only on observations, this is based on experiments, and this is based on modeling. And I must admit that my fancy things on Mediterranean fish, it's only observation, nothing else. If I try to predict my fish according to socio-environmental conditions, it's only observation, only relationship, there is no causal effect. What I can do, the best I can do with deep learning models is that to remove some layers, I can remove the MPA layers, I can randomize it to see whether I will lose some predictive capacity. So for instance, in the MED, I can attribute 10% of my explanation to the presence of MPAs. It's not causation. It's just a link between my MPAs and my level of biodiversity. There is no causation at all in all what I did in marine science. So I have a new project now because the goal is to translate this benefit of marine protected areas for biodiversity into benefits for the poorest people, to know whether we can alleviate poverty, improve health in the countries where they are the most needed. So this is my second step of Pearl equation. Pearl causation is, can we see some improvement after the creation of MPA? So it's my new project called Blue Africa. I got this big European grant to study whether the blue economy interventions has improved wealth in Eastern Africa, Tanzania, Mozambique, and Madagascar over the last 30 years. So the goal is go to the past and to know whether there's MPAs or there's blue economy intervention had an impact on people. So my core hypothesis is that since we had some desertification, climate change in these countries, the land is not so fertile now. The villages having some managed fisheries, MPAs, having some aquaculture, ecotourism, has better alleviated poverty in such conditions, trying to take advantage of marine resources at a time when terrestrial resources are posing some problems. And my main breakthrough is to have a causal relationship over 3 decades, to go back to the past and try to see a real impact before the creation and after the creation of a blue economy intervention. So the golden standard to show that kind of effect, of impact, are random controlled trials. They have been very famous since the Nobel Prize of Esther Duflo in Africa. She was one of the first to build up experiment in Africa to test whether we can invest in some treatment, in some social actions to improve the conditions of African people. So if we want to follow the path of Esther, we'll need simply to consider many villages, to split these villages in 2 groups, some receiving MPAs, some over not having MPAs, waiting 10, 20 years, and to see whether there is some improvement in terms of health or income for these two sets of villages. But this kind of experiment is posing many problems, ethical problems. We cannot set up MPAs in some villages and not in the others. And it's a lot of time. We need to wait perhaps 10 or 20 years before seeing the results. It's a shame because the causal effect for me will be the difference in incomes between these 2 sets of villages, the one with MPAs and the one without MPAs. To be a dream project, I cannot do it. So what I will do is that the solution is to use these models, these famous deep learning models to try to estimate poverty in Africa in the last 30 years. How I can do that? The same as for zebra, the same as for pneumonia, the same as for orchids. Because we know we have some DHS program making questionnaires in Africa. Now we have 100,000 questionnaires in many villages in Africa since the 90s. And what I was able to do is that to take images of these villages from Landsat Sentinel. And there is a very strong link between the image of these villages and the level of poverty. So using this deep learning model, I am able to predict the level of poverty 30 years back in all villages of Africa. So since the creation of MPAs occurred 20 years ago, it's fine, because I can go back to 30 years ago, choose my villages, and look at the trajectories to know whether villages close to MPAs are in a better shape now than it was before the creation of MPA. I'm close to the end. So the goal is to select control and treated villages. Treated is good because they are close to MPA, they are close to MPA creation. Control similar villages, same language, same people, same size before the treatment. And I will just look at the difference between the villages during years, during 30 years, to know whether there is an impact of creation of MPAs. So it's a major societal impact because we can motivate or unlock blue entrepreneurs in Africa to try to create aquaculture, ecotourism, or manage in a better way their coral reef for fishing outcomes. It's a big scientific impact because it's hard to have such long time series in social science. It's a 10-year vision because I strongly believe that there's high resolution images will change the way we monitor human well-being and biodiversity in the coming years. It's what I want to collaborate with Jessica as well on these different aspects. Because new satellites are launched almost every month with higher capacity, so we get photos everywhere at any time, so we can monitor now most of Earth and the ocean. The last one, I will be very fast, counterfactuals. So what is a counterfactuals? It's an imagination of another scenario. So for instance, we published that paper some weeks ago. We got many feedbacks on that, where we modeled the level of fish biomass on coral reefs everywhere in the tropics. And we tried to assess the gain of biomass due to conservation efforts during the last 30 years. I will skip the technical stuff, but simply we try to model fish biomass according to the level of protection. And the counterfactual is that if we had no protection, what will be the level of biomass? And when we get these two estimates, my real gain, my conservation gain, is the difference between fish biomass we have now and fish biomass without any conservation effort. It's my counterfactual scenario. It's the opposite. If we have done nothing, what is the gain? And the result we got is 10%. I was very depressed because 10% is not that much. But it means that if we still do comparison between inside and outside, we miss something. Because most of the time when we choose an MPA, it's not randomly. We choose a good site or a poor site. We choose a certain habitats. It's not very fair to make comparison between inside and outside. What is much, much, much better is to say, if the site inside MPAs was not protected, what would be the level of biomass? It's what we do with this last stage of modeling. And the good thing is that with a seascape approach and imagination scenarios, we can play a lot. So for instance, we know that we can see the boats from satellite images. We can see fisherman tourism. We can detect all these boats using artificial intelligence. And we know in the Mediterranean Sea, these pictures of the boats are the main predictors of fish biomass, much better than marine protected areas. And we can imagine counterfactuals based on that. And my tools are very good at saying, okay, what else if we had less boats? If we tend to restrict fishing boats, if we restrict the size of the boats, the noise made by the boats, the number of tourists, what can I expect? It's my scenario. So I can put that in my model. So instead of having the picture here, I choose this one and I see my outputs. The same thing, if I authorize more boats, what can I expect? So we can imagine with stakeholders many scenarios. What do you want? Is it a world with co-benefits? Is it a world with more benefits for nature and less for people? Is it a world for more benefits for people than for nature? So I will stop here. Thank you for your attention. I'm sorry for my terrible French accent.